Histology is generally thought of as the study of tissues. Specifically, you're looking at all the different cell types and cell shapes that make up your different tissues, how they're arranged, your cellular arrangement. Are they packed tight together? Are they in rows? Or how are they joined together? The connections between cells. About extracellular materials. So in your bone, for example, most of your bone is not made up of cells. There are cells scattered through your bone, but most of the bone is extracellular material, that hard material of the bone. The same would be true for a cartilage. For blood, for example, it's mostly fluid in there with cells in between. We can think of it as the microscopic study of organs and tissues, what things look like under the microscope. And when you do that, when you look at these organs and tissues under the microscope, you're looking at these cellular arrangements, what kinds of cells, shapes of cells, and so on, or extracellular materials make up those structures, the organs and tissues of the body. The next concept I want to introduce is cell lineage. Cell lineage. So we start with the zygote. Our first cell, it divides, we get more and more cells. As we get more cells, we eventually get the germ layers to form, the endoderm, ectoderm, and mesoderm. And then those regions form more specific types of tissues. So you have the regional specializations. So cells in one part are becoming one kind of structure, and cells in another part become another type of structure, and so on. So, for example, here we have a cell of the ectoderm. As the cells divide, some of them become identifiable as surface ectoderm cells, as opposed to extraembryonic membrane or neural ectoderm or neural crest. So we have surface ectoderm. Some of those cells that are forming the surface ectoderm may eventually form into hair follicles. In there, they fold in and go deep into the dermis. Some of those down in there might form the hair matrix. Hair matrix cells divide and make more hair matrix cells, and some of those matrix cells will eventually form the keratinized cells that make up your hair and inner root sheath of your hair from your hair matrix. So things become more and more specialized. These are referred to as determinate steps. Their fate is more and more determined. So there are genes that are turned off or turned on during these cell division processes in each of these determinate steps. So some genetic change here caused that surface ectoderm to become part of a hair follicle. The hairs, these cells of the ectoderm started to change in such a way that they became, started the root of the hair follicle to turn into the dermis, and then some change within some of those cells caused them to become hair matrix cells. Some genetic change, a determinate step, a gene turned off or turned on to facilitate the behavior of that cell in that tissue. Most of these determinate steps are one way. There's no turning back. So once you've made the change, you can't go back the other way. It would be hard to take a hair matrix cell and then stick it into a muscle and assume that it would function as a muscle cell. You can't put it in the blood and think it's going to act like a blood cell. Once they go to the next stage, the next determinate step, their fate is more and more locked in. They can't go back the other way. The next important concept coming from the idea of cell lineages and how that cells become more and more specific. These terms, totipotent and pluripotent, we'll often run into with regard to what we call stem cells. Cells that are dividing and can have a number of fates down the line. If a cell is totipotent, that means that all of the different cell types of the organism can be derived from that cell. The zygote is considered the ultimate stem cell. As the zygote divides and becomes like 2 cell, 4 cell, 8 cell, some of those cells that divide from that original zygote would also be totipotent. You can grow a whole new organism from some of those individual cells. You can isolate them, they start to divide and eventually form an entire organism. So those cells are totipotent. But fairly rapidly in the early stages of division of the zygote, you're going to get some specialization of the cells. Some of the genes will be turned off. They will no longer have the ability to become all types of cells in the organism. These are referred to as pluripotent cells. Then, if they can't become anything, they might not be able to become anything that's associated with the nervous system, for example, at a certain point in the developmental sequence, or they can't become a gut cell in the developmental sequence. So these are still stem cells. They have a lot of different cell types that might be able to be derived from them. That's where we get the plural, and many different things. But they have a limited potential. They can't become any type of cell in the body. Pluripotent cells. And then eventually these cells become more and more specific and finally become what we call fully differentiated cells. Fully differentiated cells, they can't change to become something else. Sometimes these are referred to as terminal because they no longer divide any further. So they reach a point where they are the last cell in their lineage. They will not undergo another division to form more cells of the same type. So we have some of our body cells are terminal cells. Many of our neurons, our muscle cells, and so on are terminal cells. And then there are some that can produce more cells of the same type. They still are fully differentiated cells, but they will produce more cells of, say, skin cells, lining of your oral cavity, places where you see a turnover in cells frequently in some connective tissues and so on. These are fully differentiated cells, but they still are undergoing cell divisions to make more of the same cell type. So these are different stages, totipotent or pluripotent or fully differentiated. Now that we have a little bit of background on the development of the different tissues, that specialization process whereby we get these specific cell types, let's talk about our functional classifications, our groupings of tissues. There are four primary categories. First of all, the epithelial tissues. When we think of epithelium, we think of our skin first off. So we know that's a kind of covering. So in a definition of epithelium, we would talk about coverings and linings. So they would line your oral cavity, your nasal cavity, and all through your gut, inside of your blood vessels, and all those things. So forms coverings and linings. In addition to that, to be an epithelial tissue, it also has to rest on a basement membrane. Rests on a basement membrane. We'll talk about that basement membrane here a little bit later. It's an extracellular layer. It's not made out of cells, not made out of the actual cell material, but something secreted by the cells. It forms a basement membrane. It's kind of a glue that holds the epithelium in place. And usually, because it's a covering or lining, this is associated with the free surface. A free surface. So of course your skin has a free surface, but any other cavity in your body would have a free surface side to the epithelium and then a basement membrane side to the epithelium. So this kind of sets up a polarity in any epithelial, a basal portion of the epithelium and a free surface layer of the epithelium. So anyway, so these are epithelial tissues. Occasionally, a tissue that begins to form as an epithelial layer as part of one of these coverings or linings will migrate away from the surface and embed itself deeper in the tissues where it's no longer associated with a free surface. And it's no longer part of a covering or lining, so it's a dissociated type of epithelium. In that case, we call them epithelioid tissues. They had an epithelial origin. They were part of an epithelium, no longer associated with the free surface. So that sets our epithelioid tissues apart from some of our other tissues here. So portions of your adrenal medulla, for example, would be your pituitary gland is a really good example of epithelioid tissue. It was at one time part of the roof of your nasal passages in there in the oronasal cavity, and then it migrated away from that position. So there are a few organs in the body that are epithelioid. Started as epithelium, but then moved away from that. We will break down the specific types of epithelial tissues here in just a little bit. The second group are the connective tissues. The connective tissues usually have some kind of a function to join two parts together. That's one possible role. Join structures together. Sometimes they simply fill space. Between structures, they just kind of take up the space in there, so they can be just space fillers, and sometimes they give an overall support or shape to a structure or organ. Very important, of course, in your skeletal system and cartilages and so forth. We have that support mechanism in there, joining bone to muscle. We need that strong support in there. So anyway, these are connective tissues. Included in here, not doing any of those things, but for a number of reasons, blood is included with your connective tissues. Okay? And later on we'll break down all the different types of connective tissues. Now on to our next. Muscle tissues. This is fairly simple to think about what muscle tissue is. We know it's in control of movement. Both our external bodily movement, moving our arms around, our facial expression, doing those kinds of physical movements. So it's responsible for movement. So it's both external, what we've just described as mainly externally visible types of movements, but also internal types of movements, things going on inside your body. It includes all sorts of different things to have cells contract that help to move sweat to the surface of your skin. Cells that contract to cause peristaltic movements through a tube of the reproductive system, the heart, and so on. There are three kinds of muscle tissues. We're going to break them down later on, but I'll go ahead and list them briefly here. What we call skeletal muscle accounts for our voluntary movements, and most of everything connected to the skeleton is part of our voluntary movement control system. So skeletal muscle. And then cardiac muscle, only found in the heart. Cardiac muscle, the heart muscle. And then the remainder of the muscle is what we call smooth muscle. It's found in most of your hollow visceral organs. Those are the organs inside your body that have an empty space in them, some kind of a tubular passage, or like your urinary bladder, a cavity on the inside. So in your visceral organs, also along your blood vessels, you have smooth muscle controlling the diameter of your blood vessels that the blood passes through. Also inside your eye, the focusing mechanism for your eye and the pupil dilation. That's all controlled by a smooth muscle. And then finally in your skin, there's what we call arrector pili muscles that cause your hairs to stand on end. Your goosebump muscles in your skin are smooth muscles. None of those are voluntary things. We don't have any voluntary control over those. No voluntary control over our heart muscle either. So skeletal muscle, cardiac muscle, and smooth muscle, we'll look at each of those in detail later on. Our fourth tissue category, and this is the last of our tissue categories. We've been through epithelium, connective tissue, and muscle. Nervous tissue is the fourth category. Nervous tissue includes a number of cell types, not all of which are conducting nervous impulses. We kind of connect everything that is associated with the nervous impulse conduction in this category, so it's easy to define what is a nervous tissue, but it includes more than what you might think. So these are associated with neural impulse conduction. Now, when we studied embryology, we saw the development of the neural tube. So everything derived from neural tube is going to be considered a nervous tissue. Also, some of the tissues that come from neural crest, so some neural crest tissues are also included in this category if they are part of a neural impulse conduction structure. So things included in this include neurons. Those are the things in the central nervous system that actually convey the neural impulse. Then associated with neurons, we have a number of what we call neuroglial cells. That are there to support the neurons. And we're going to list those later on. It includes astrocytes, oligodendrocytes, microglial cells, and the ependymal cells. So 4 different categories of neuroglial cells there. Also in our peripheral nervous system, we have something similar to the neuron, and sometimes they're referred to as neurons. More technically, they're referred to as ganglion cells. The ganglion cells. So they function like neurons in the peripheral nervous system. And surrounding some of the ganglion cells, we have what are called satellite cells. Satellite cells, they kind of pack in around and protect the ganglion cells, satellite cells. And we have the Schwann cells that form around the outside of nerve fibers. And we'll describe how that works later on as well. So this kind of finishes out our different tissue categories. We've seen epithelium, connective tissue, muscle tissue, and nervous tissue. Nervous tissue, things associated with impulse conduction derived from either the neural tube or the neural crest. Now we're ready to go into more detail on each of our tissue types. We're going to begin with the epithelial tissues. And before we start describing each specific epithelial tissue type, I will make a list of the different tissue categories, types of epithelial tissue. These are organized according to shape and whether or not they occur in a single layer of cells or if they're multiple layers of cells, what we call stratified. Simple if it's a single layer, stratified if it's multiple. Now the shapes, squamous, so the first category, simple squamous. Squamous means flat. It's a flattened cell, so a single layer of flat cells. Would be simple squamous. Several examples of that in your body we'll talk about. Next would be stratified squamous. Here you have many layers of cells, several layers of cells stacked one on the other, and only the top layer has to express that squamous shape. The ones down at the base of the layer near the basement membrane don't need to do that. Next is simple cuboidal. Cuboidal doesn't mean they're a perfect cube, it just means that they're approximately as tall as they are wide. Their length and width dimensions makes them roughly cuboidal. They look more like square pillows or something like that than actual cubes. They are sometimes low cuboidal, where they're a little bit shorter, or sometimes they're a little bit taller, but roughly cuboidal, roughly as tall as they are wide. Simple cuboidal epithelium. Occasionally you'll see stratified cuboidal. Epithelium in some parts of the body, mainly your sweat ducts and your salivary ducts is where you would find those. Stratified cuboidal. Next, columnar cells, simple columnar. Are much taller than they are wide, and they look like columns. Simple columnar epithelium. It's very rare to see a stratified columnar epithelium like it was for stratified cuboidal. About the only place you find those are in the largest of your sweat duct, not sweat ducts, your salivary ducts. Your largest salivary ducts have a stratified columnar epithelium. Now, we've taken care of most of our different shapes here, but there's a kind of epithelium that's referred to as pseudostratified columnar. It looks like maybe it's made of multiple layers of cells, but in fact all of the cells make contact with the basement membrane and extend up from the basement membrane of varying heights, and they squeeze together to make them look like multiple layers of cells when they're not. So pseudostratified columnar epithelium through most of your respiratory system. You can find some of that. And let's see, the last category is what we call transitional. That means it can change its shape, mostly rounded cells, but they can flatten out somewhat and be stretched and change. So they're called transitional. Those are our 8 categories of epithelial tissues, 8 categories of epithelial tissues. So now we'll look in detail at simple squamous epithelium. Simple squamous epithelium is, of course, a single layer of cells. It's simple, not stratified, not multiple layers of cells. Squamous means flat. It's an epithelium, so it's going to be on a covering or lining and have a basement membrane. I always draw the basement membrane in orange, so my orange line here will represent the basement membrane. And then on top of that, I'll draw in my different cells. Usually I make the cell outline in red. Use a red marker to do that. So here's a simple squamous cell, here's another simple squamous cell, and another on here like this. So a single layer of cells on a basement membrane. The top would be the free surface, and in these cells you'll have a nucleus, and it's fairly common to see your nucleus kind of squished flat in the cell, conforms with the flatness of the cell, something of that sort. So that's simple squamous epithelium, a single layer of flattened cells. Places where you find the simple squamous epithelium, I'm going to go ahead and write this, get the right word. I'll start with endothelium, I think, first. Endothelium. Endothelium is the cell type that you find lining your blood vessels and inside your heart, inside your lymph vessels as well. I'll just write blood vessels out here because that's what we usually associate with this term. Endothelium, single layer flattened cells. Next, mesothelium, associated with mesenteries. That's where the MES comes in, mesenteries. Mesenteries and membranes that cover your organs and line your body cavities are mesothelial layers. So, put here, line body cavities. That means like your abdominal cavity, your pleural cavities that have your lungs, and your pericardial cavity where your heart is found. Those are the kinds of cavities we're talking about there. And cover the organs within the body cavities. Now, many of your organs are connected to each other or to the body wall with a sheet of material, a very thin layer of material, a very transparent layer of material in there that's simply called a mesentery. So I'll put here mesenteries. So those things are mesothelium. Third category, inside your lungs, where you have that interface between your air space of your lung, lies right next to a blood vessel, so we can get oxygen to travel across from the air to the blood. We have a couple of simple squamous cells in there. Of course, the blood vessel is lined with endothelium, but in the lung we have another type of flattened cell. We usually refer to it as a type 1 alveolar cell. Type 1 alveolar cell, that flattened cell in the alveoli, that's the little airspace, small air sacs inside the lung. They are lined with type 1 cells, simple squamous cells. One more location where we find this, when we study the kidney, we're going to study a structure called Bowman's capsule. Bowman's capsule is the place where, that cavity where fluid moves out of the blood, out of small capillaries into a space, a capsule, the Bowman's capsule. So surrounding Bowman's capsule we have simple squamous cells. Surrounding Bowman's capsule. So those are 4 locations where we have simple squamous epithelium. Now for stratified squamous epithelium, we're going to again begin on a basement membrane, and on this basement membrane we will have cells, we'll outline in red here. At the base they will not be squamous, they'll be irregular shapes, rounded or nearly cuboidal in shape. So somewhat irregular in shape, and then the next layer up and then the next layer up. And as we move toward the surface, they become more and more flattened. So as long as we have flattened cells on the surface, this is going to be a squamous type of epithelium. Again, we have a basement membrane down below, we have a free surface above, and then our layers are stacked between the basement membrane and this free surface we have here. Now, stratified squamous epithelium can occur in 2 different forms. non-keratinized or keratinized. If it's keratinized, the cells on the very surface are dead cells. There can be layers and layers of dead cells on the surface, like on our skin, especially on the heel of our foot or the palms of our hands. Several layers, many layers of dead cells there. As you go down through those layers of cells in a keratinized epithelium, eventually you get to some of these irregularly shaped cells, more flattened, but then all of these down below are living cells. They have nuclei, cells at the base divide, and as they divide, they add more cells to the next layer. And these cells, once they move to the next layer, they don't divide any further. They are just pushed up by the addition of more cells down below from that basal layer. So anyway, this thickness increases. And we'll talk more about keratinized stratified squamous epithelium when we talk about the epidermis of the skin, because that's our primary example of keratinized stratified squamous epithelium, the epidermis. Non-keratinized stratified squamous epithelium, more like what I've drawn here, can be found in the oral cavity. So lining your mouth, you have a stratified squamous epithelium that is non-keratinized. It has flat cells on the surface, and those flat cells have a nucleus, and they're not dead cells until they're sloughed off and die. Oral cavity, the esophagus, and vagina are locations where you find non-keratinized stratified squamous epithelium. Simple cuboidal epithelium rests on a basement membrane again. These cells are roughly as tall as they are wide, so they appear pretty much as square-like shapes on a basement membrane, and they have a free surface. Single layer of cells, because this is a simple epithelium, simple cuboidal epithelium. Like this, each of them would have a single nucleus to the inside. Examples of places where you find this. You find it on the surface of the ovary, in thyroid follicles, thyroid follicles are little cavities that contain a precursor to your thyroid hormone. They're like little gel packets, little bubble-like packets that are surrounded on the outside by a single layer of cuboidal cells, thyroid follicles. Your kidney tubules, that's a good one. Kidney tubules, so as the urine passes through these tiny tubes, water is reabsorbed and urine is changed as it passes through those tubes before it dumps into collecting ducts that drain into the renal pelvis. So anyway, kidney tubules. The next one would be small salivary ducts. And ducts of maybe some other kinds of glands you could think of, small ducts in the body. But anyway, these are some examples of tissue or places where you find this kind of tissue, this simple cuboidal epithelium. So next on our list is the stratified cuboidal epithelium. Again, we start with a layer of basement membrane, and on that basement membrane we'll put some cells. Remember, this is stratified, so there's going to be more than one layer of cells, and the ones at the base are not necessarily neatly cuboidal like those on the surface, but anyway, we've got some cells down here. And then we have the surface cells that are more cuboidal, more regular in shape. Like this. The locations where we find this include some of your medium-sized salivary ducts and your sweat ducts. I'm going to make another picture here in a minute that shows what a duct will look like in cross-section, not a flat view like this, but a circular view. But I'll just go ahead and write down here, medium salivary ducts, and sweat ducts, the ducts of your sweat gland. So if we were to make a cross section of one of these ducts, look, we cut through the skin, this is the way we usually see it histologically, we would see a circular basement membrane around the outside. In the middle would be the cavity of the duct, a tubular passage, what we often refer to as a lumen. A lumen is the cavity of an internal structure. So we would have one layer of cells up against the basement membrane here. The more irregular cells. And then out toward the middle we would have a fairly even lumen space here with our cuboidal cells spaced out around this. So that would be a stratified cuboidal epithelium in a duct, like a sweat duct. In a medium salivary duct, we'd have the same kind of wall, but the luminal space in the center would be bigger on a typical medium salivary duct. So anyway, that's stratified cuboidal epithelium. Okay, so next is simple columnar epithelium. Simple columnar epithelium sits on a basement membrane, has cells that are taller than they are wide. It's a single layer since it's simple columnar epithelium. So we have tall cells in a row. Simple columnar epithelium. Now we're going to put nuclei in them. The nuclei usually are found in a fairly uniform position across the row of columnar cells. So they look like they're all soldiers in a row kind of arrangement here. Real even look to the columnar epithelium. Places where you would find simple columnar epithelium include your stomach lining, all of your intestines, your gallbladder, bile ducts. Those are some of the places where you're going to see a simple columnar epithelium. There may be some other places as well, but that's a good list. Simple columnar epithelium. Stratified columnar epithelium. The cells at the base of the epithelium are not columnar at all. They'll be irregular small cells. And then above that you'll have the columnar shapes, usually very even at the top. Usually just two layers like this. Places where you find this, there's really just the larger salivary glands, larger ducts of salivary glands. May occasionally be seen, and as far as that goes, also stratified cuboidal. Sometimes comes in the interface between transitional epithelium and stratified squamous epithelium in the urethra, but it's very irregular and not a good place to go looking for it, actually. So anyway, this is predictable. Large ducts of salivary glands will have this stratified columnar epithelium. Pseudostratified columnar epithelium. All of the cells in the pseudostratified columnar epithelium have contact with the basement membrane. Not all of them reach to the top of the surface layer. So you get a whole bunch of very irregular shapes, and their nuclei are situated in different positions in different cells. So it looks like it could be multiple layers of cells across here, but the nuclei are just in different positions in each of the cells. And like I said, some of these cells do not reach all the way from the basal membrane to the surface. So this gives us this pseudostratified look, falsely stratified look, pseudostratified columnar epithelium. Places where you find this, there are actually several in the body, but we associate it mainly with respiratory passages. So olfactory epithelium, where you have your sense of smell, is this pseudostratified columnar epithelium. What we generally refer to as respiratory epithelium. Lines like your sinus passages, your nasal passages, and then once you get into your windpipe and into your larynx, your trachea, and your primary bronchi, secondary bronchi, all of those passageways have that pseudostratified columnar epithelium. It's not uncommon in some of the male reproductive system ducts, so the ductus deferens. Has a pseudostratified columnar epithelium. So anyway, it's usually ciliated, has cilia at the top, and all of the respiratory epithelium that helps to move particles out of your lungs or away from your mucous membranes and combine them with the mucus so that it will be swallowed, all that debris, particles, dust, and so on. So that's pseudostratified columnar epithelium. Next, transitional. Our final epithelial type. Cells rest on a basement membrane. Transitional is stratified. It has multiple layers of cells. It's not called stratified transitional, I guess, because there's not a simple transitional. You have a number of smaller cells down at the base of transitional epithelium. And then as you go up, the cells get a little bit bigger. They're all pretty much rounded cells. The nuclei in each of the cells is about the same size as you go from the bottom to the top. But because the cells are smaller down here in the lower layer, it looks like the nuclei are a little more crowded. And then when you get up here in the higher levels, the nuclei seem to be more spaced out. Not as tightly packed in the upper layers as they are in the lower layers of the epithelium. That's one of the easy identifying features for transitional epithelium. Also the very rounded cell shapes at the surface. Now it's called transitional because it can stretch. This is found in the urinary bladder. So when the urinary bladder enlarges, these cells all flatten out a bit. So this is the relaxed state. We'll call this relaxed. Sometimes it's called contracted state. And if we were to distend it, to stretch it, our cells at the bottom would do some stretching, but each of them proportionately would not have to do quite as much stretching as those that are toward the surface. So in the stretched state, the transitional epithelium has thinner layers all the way around there, but the cells at the surface are a little more flattened. They're not squamous. They're just not as round as they were before. So they undergo this change in shape. They're transitional. I've already mentioned that these are found in the urinary bladder. By the way, this is the stretched or distended type. Urinary bladder. And really it's only found in the urinary system. So as you go from the renal pelvis, really inside the kidneys where this starts, in the renal pelvis, you have that extend through the ureters that lead down to the urinary bladder and then into the urethra through most, but not all, of the urethra. Contains transitional epithelium. So that's the last of our 8 different epithelial categories, transitional epithelium.